All right, welcome everyone. I'm Pangkong Fox, the Science Engagement Director for the CACNA1A Foundation, and I'm here with Dr. Jen Pan from the Broad Institute of MIT and Harvard, who is being awarded a $50,000 seed grant, research grant from the foundation for her project that is titled Elucidating the Cellular Deficits Underlying the Missense Variants of CACNA1A Identified in Patients. Dr. Pan is the Director of Translational Biology at the Stanley Center for Psychiatric Research at the Broad Institute. She received her PhD in neuroscience from Brown University, and her research has been focused on studying genes implicated in psychiatric disorders and developing next generation therapies to treat psychiatric disorders and neurological disorders. So welcome, Dr. Pan. Thank you for being here to talk about your work on CACNA1A related disorders and how this will impact our CACNA1A community.
Hi, Pangkong. Hi, thank you. I'm really excited to start this conversation about the project and looking forward.
Great. Okay, so before we discuss the specific research project, we'd like to know how you actually came into this work. So could you briefly talk about how you became interested in studying CACNA1A?
Oh, sure. I did my PhD thesis in a close analog of CACNA1A, which is CACNA1B. I was interested in the molecular function of CACNA1B for my PhD thesis. And fast forward many years, I currently work at Broad Institute, which is a leading institute in human genetics. When I encountered it again in the past few years, during the project for the epilepsy without the walls, and when I heard that many missense variants of CACNA1A have been found in kids with epilepsy and other neurodevelopmental disorders, I was very keen on understanding how human DNA change causes dysfunction in this CACNA1A gene.
Great. You've spoken at our family conference, our family and scientific conferences in the past. You spoke in San Antonio, Texas in 2022, and then this past summer in Bethesda. How has working with the foundation and meeting families in our community impacted your research? How does this make you think about developing treatments for CACNA1A related disorders?
Right. It's really interesting. I have to say that these meetings that's organized by CACNA1A Family Foundation are my favorite meetings to go to. It's not very often that we, as scientists sitting in the lab, can see the impact of the work that we pursue at the bench in the real world, to see that real world impact. I have a good reason to be interested in CACNA1A, mainly from an intellectual perspective, disease perspective, but working with the CACNA1A Foundation and meeting families really brings that real world meaning and impact to my research. It makes me feel more urgent and makes me feel more aware of the meaning of our scientific research. I wanted to help with people who harbor the CACNA1A mutations and wanted to contribute to finding treatment for these people who have the mutations.
That's really wonderful to hear. And I think a lot of our families will appreciate that too. I think for them, meeting scientists for the first time is very, kind of intimidating. But hearing that meeting them is so impactful to you as a scientist and to your work. That's really important for our families to know. And this is why it's so important for us as a foundation to bring both the scientific community and the patient community together so that you can both influence each other in that really positive way.
Absolutely. Meeting families are in the past year to the families in the foundation who brings these families together. It's really made impact. Make me look a little differently on the research I do. It really does.
Well, that's a good segue into my next question. Let's explain to the families exactly what you're doing with this research project. So can you summarize what your project is about and how you envision this work moving our community closer to better treatments?
Absolutely. The objective of this research project is to functionally characterize a set of novel CACNA1A missense variants that are found in the patients of neurodevelopmental disorder but have not been characterized. We will first categorize them into loss of function or gain of function, but then we'll focus on those loss of function mutations and categorize major cellular and molecular deficits. For example, we wanted to understand when you have a particular loss of function variant, does this variant reduce the amount of the mRNA produced? Does it make the protein to be degraded or not made? Or the protein is made but unable to traffic to the surface? Or the protein is made and trafficked to the surface, but unable to function properly at the plasma membrane. I think distinguishing these different aspects of the deficits is going to have an impact in how we think about treating them and mitigating these dysfunction. So the result from this proposal, together with our past result, probably is going to hopefully serve as a first step in obtaining the electrophysiological profile for a large number of human CACNA1A variants, use a consistent analytical method so that we could start building a functional map at the molecular resolution. Furthermore, I'm just really excited that we're going to be able to dissect different potential molecular deficits of loss of function variants. I think this will lay the strategic foundation for the therapeutic approach to normalize the deficits of these loss of function variants.
No, that's so wonderful to hear. And I just want to, remind the families out there that, if you think about these neurons, right, they're really, really long. And there's one part of the neuron called the cell body, and that's where proteins are made, or any of these molecular tools are made. And then they have to eventually get to where they're needed. And in the case of CAF2.1, which is this calcium channel that's encoded by the CACNA1A gene, it has to get to the cell surface at the very end of the neuron. And so when we talk about these trafficking defects, there's this idea that loss of function could be because of the channel not being able to open correctly, but could also mean that it's not able to get from where it was made all the way down to the neuron. Or maybe at some point it was degraded by the cell because it just wasn't a functional protein. And what Dr. Pan's work is going to do is to really go in and figure out what is happening at that molecular level. And once we understand what that is, we can start to think about treatment approaches that can really target those specific mechanisms, so we can have really nice precision medicine.
Exactly. I think this level of cellular mechanistic understanding of the impact of human variants will give us, and then also just a distribution of how many at the population level, how many of these variants are each of those categories, gives us a sense of in general what to expect. I think those are going to be very helpful in thinking about laying some level of strategic ground to thinking about how to fix each of them.
Yeah, and that's eventually that's the big goal that we would like to bring to our families in the end. So I know you spoke a little bit about what was most exciting, but again, any other things that really excite you about this project and continuing to work with the foundation and the families?
Yeah, so this project builds upon our previous results of characterizing about 50-ish de novo variants and population variants. And I'm confident we'll be able to we'll characterize additional missense variants of CACNA1A in this funded by this project, and build a better map from genotype to molecular phenotype correlation based on this work. But in particular, as we just discussed, and I think what this project means to me is that in addition to characterizing, we'll start to understanding potentially different types of deficits, even in one loss of function or gain of function category. So we can start to build more precise strategies to treating each one of them. And then that would give us a lot of confidence in downstream strategies in coming up with small molecule or other ways to mitigating their dysfunction. And I'm just in general talking about excitement. I'm excited about going to another conference of the family, CACNA1A Family Foundation and community. Your energy really inspired many of our scientists at the bench.
All right, thanks for that great summary. So Dr. Pan, on behalf of the CACNA1A Foundation, I would like to thank you for your dedication to our community, especially that original characterization of those 50 variants. That was an initiative that you pursued independently on your own for the last few years, and we didn't support that. So we're just so grateful that you did that, and then now we're able to fund the rest of that work. And again, just to our families, this work is so critical to understand what treatments are going to work for specific variants, and really understanding those disease mechanisms is how we're going to get there. So thanks again, Dr. Pan. We really look forward to how these results are going to shape treatments for our community.
Thank you so much for funding this work. I'm really looking forward to working with you to move the understanding of CACNA1A and potentially the treatment strategy down the road. Thank you.